Since Darwin, there has been interest in the idea that there may be more variability between males of a species than between females. Empirical studies have typically shown greater male variability, especially for morphological and cognitive traits in mammals. For example, male red deer are more variable in size, male guenons have more variable skull sizes, and male chimpanzees have greater variation in brain structure. In humans, males show greater variability in birth weight, adult weight, and height, body mass index, and brain structure. Greater male variance has also been observed in performance traits such as sprinting speed and various measures of muscle strength. Yet, outside of a few limited research areas, the study of putative differences in variability between the sexes has rarely been conducted. However, new work by Zaticek et al. On mice casts doubt over the ubiquity of greater male variance in mammals. While Zaticek et al. Found more variance in males for morphological traits, they found no systematic difference in trait variance between the sexes for physiological, behavioral, and hematological traits. Moreover, they observed more variance in females than males for eye traits and immunological traits. Perhaps then, whether there is greater male variation in traits between the sexes depends on the trait type in question. Is energy expenditure predicted to exhibit greater male variation? Energy expenditure represents the metabolic costs of multiple physical, physiological, and behavioral traits, which could result in energy expenditure exhibiting particularly high, greater male variation through a cumulative effect if those traits mainly exhibit greater male variation themselves, or a lack of greater male variation if many of them do not. To test for greater male variation in energy expenditure, we analysed a large database on energy expenditure in adults, the IAEA DLW database representing 1,496 men and 3,314 women. Remarkably, we found that, even when statistically comparing men and women of the same height, age, and body composition, men exhibited far more variation in total, that is daily, energy expenditure, far more variation in energy expended specifically on activity, and on basal energy expenditure than do women. Considerable greater male variation in both total and activity energy expenditure can be explained by greater male variation in levels of daily activity, which is documented in the literature. The large greater male variation that is present in basal metabolic rate is remarkable. We argue, with support from the literature, that it may be explained by greater male variation in the size of energy-demanding organs, most notably the heart, lungs, brain, and kidneys. There is also evidence that mitochondrial energetics in response to low metabolic demands vary more in men, as does the abundance of different mitochondrial proteins in skeletal muscle. So, our study has uncovered a very large effect size of greater male variability in human energy expenditure. There are several potential explanations for this. However, given that men have experienced greater reproductive variability than women throughout most of our species' evolutionary history, including the present, and that total energy expenditure variance decreases with age more in men than women, one plausible mechanism is that energy expenditure is directly or indirectly under sexual selection. If you'd like to find out more about this research, you can access our paper published in the Journal of Human Evolution at the journal's website, or contact the corresponding author.